Hi, this is Miss Litton, and this is my wonderful Period 3 AP Biology class. Say hi.
Hi.
All right. So we're going to discuss beyond Mendelian inheritance, but I just want to back up a little bit, make sure we're all on the same page. So Mendel had, we learned about this last time, 3 laws. Okay, so let's do them like logically in order. Which one would we say first? The law of dominance. Good. And that would mean that, an example with that would be like when a tall plant, if you inherit 2 alleles, one for tall and one for short, that a tall pea plant would be dominant over a short pea plant. It's not like it's tall, medium, and short. You're either tall or short, okay? And so a dominant allele can mask a what? Recessive allele. What is an allele?
Variation of a gene.
Yeah, a variation on a gene, okay? And so far we've only talked about having the combination of only 2 choices there for our allele. We've had big T and we've had little t, or big R and little r. Okay, that would be in simple dominance, but we're going to upgrade that today. Okay, so law of dominance, what's our next law? Law of segregation. And the law of segregation says that each gamete will only inherit what? 1/2 of the homologous pair. So if you have something like a heterozygote with R's, or widow's peak, right? Because widow's peak is dominant over straight hairline. If you're a heterozygote for it, some of your gametes would get the allele for widow's peak, and some would get the allele for a straight hairline. Okay? When we talk about the next law, what is that? Law of independent assortment. We need to bring in 2 sets of alleles to talk about independent assortment. And we're saying they would be on 2 different chromosomes. And you can get any combination in that. And in the example we looked at were peas, and we said they could either be round or wrinkled, and they could either be yellow or green. And we talked about when you form your gametes, that depending on how they line up during metaphase 1, that you could get variations in your gametes. And we said, for instance, you could get the allele to be round with the yellow color, or you could get the allele to be round with the green color. Okay, law of independent assortment. So now what we're going to do is we're going to go beyond Mendelian inheritance and look at some exceptions to those rules. And one of those exceptions is multiple alleles. Now I want it to make sense to you. I don't want you to be having to memorize this. Just make it be logical to you. So multiple alleles, okay? When we know what alleles are, that's an alternative form of a trait, right? So in this case, we've just had big T and what? Little t. But now we could have multiple capital T's. So it's not just large and round or wrinkled, but we could have variations on it. And this happens with blood type is a really good example of it. There are 3 different alleles for blood type. Okay, so big IA, big IB, and little I. Now you still have the law of dominance, because big I is going to be dominant over what? Little I. But now we have more. Before we had 3 different genotypes we could form, right? Big T, little t, and little t, little t. Now we have several different genotypes that we could have. So I want you to think if this is the phenotype, type A blood, you still only have a homologous pair of chromosomes. So you can't get all 3, right? Unless non-disjunction took place, right? You only get 2. Which 2 copies would you need to get in order to get type A blood? Discuss with BioBuddy. You have 3 to choose from. Which 2 would you need? Okay, what are all your options that you would have to have in order to get type A blood? Is one of them a homozygote and one of them a heterozygote? Okay, so you could get big IA, and be homozygous for type A blood, or big IA, little I. Big I is still dominant over little I, right? And you could be a heterozygote for type A blood. Now, what are your options for type B? Yes, same as before, right? Okay, and how about AB blood? Yeah? Okay, now this is another exception to the rule. This is an example of codominance. It's where both alleles are expressed. So on your red blood cells right now, if you are type A, which I believe I am, then all the antigens on all my red blood cells say type A. If you are type B blood, they would all be B antigens. They don't look like B's, that's just how they signify them, okay? If you are AB blood, you could have A antigens and B antigens. What do you think type O blood is? No antigens. Okay, and what would be the genotype for no antigens? Little I, little I. Okay, so when you talk phenotype, that's when you say A, B, A, B, O. When you talk genotype, these are your actual combinations of alleles. But we don't leave blood type there. We say A positive and A negative. That is another set of antigens, RH antigens. If you are A positive, you have A antigens and the RH antigen. If you are A negative, you only have the A antigen. Okay? And if you are O negative, you have no antigens. If you're AB positive, you have them all. And there are other antigens besides that. You just don't need them to know about them for blood transfusions. There's M's and N's also. So there's other antigens on your blood cells. Now, the concern will be about when transfusions take place and what you can accept, right? And basically, you can give blood to anybody whose immune system you won't piss off, okay? So this is how it works. If I have type A blood, I will not, even we haven't talked about the immune system yet, you have things called antibodies that tag antigens, okay? So if I have type A blood, I will not make anti-A antibodies, because if I did, I would be attacking my own blood and causing it to clot, and then I would die, okay? But I could make anti-B antibodies. Now, I have never had a blood transfusion before, so I'm fairly confident that I don't have any of those antibodies in my blood because I've never been exposed to type B blood before. So in a pinch, if I needed some blood, I actually could get a transfusion of type B blood. But the second time, it would kill me because I would have memory cells and I would have antibodies that remember B blood and they would attack and destroy it. Now, where could I have been exposed to B blood is when I was pregnant. I was pregnant 2 times. Let's get Bad Choice Barbie. Okay? If Bad Choice Barbie, okay, has type A blood and her baby in here has type B blood, oh, lifting up her dress, okay? If her baby, look, I embarrassed him. It's a Barbie, okay? If her baby in here has type B blood, for those of you listening in, just Google Bad Choice Barbie. No, I don't know what it's called. No, don't Google it. Don't Google it. Okay. So if her baby has type B blood, then during the birthing process, things can get a little rough because the placenta, red blood cells and white blood cells don't cross the placenta, but during the birthing process, some things tear. It's a little messy in there. She could be exposed to some type B blood. So her body will react to that type B blood. She will build a whole immune response, and she will have memory cells that remember B blood. So if she gets pregnant a second time, and that baby also has type B blood, come on, get your uterus going here. And that, that's exactly what would happen, okay? So if she gets pregnant a second time and that baby, she has memory cells, she has antibodies that will cross the placenta and go in there and clot the baby's blood, and that's super sad. I know a woman between child 1 and child 2, she had 2 spontaneous miscarriages, and it's usually kind of far on in the development because she had developed an immune response to that. Now they protect against it. So if you get exposed, they check the baby's blood and your blood as soon as you're born, and if that baby's blood is different where it would trigger an immune response, they give you an immunosuppressant, usually stay like maybe an extra day in the hospital or so, in order to make sure that doesn't happen. They check on your first child. Yes?
Is that how how the woman would also die during pregnancy too?
Well, women can die for various reasons during pregnancy. I would say probably a common one with I mean, I don't know how common it is, but it's from hemorrhaging, from blood loss. darn you, baby, would be from blood loss. That would be why women die in childbirth, because the baby's not exiting correctly, or they have to do an emergency surgery, and there's some complication with that. Okay, so it's not going to hurt her. It's not going to hurt the mom that she has the baby that's in there that has it wrong, but it would hurt the baby. Okay? All right, now keep that choice, Bobby. So let's take a look here. Okay. And I would like you in your practice problems, I would like you to cross a heterozygote A with a heterozygote B and look at the possibilities for the children's blood type. Heterozygote A with a heterozygote B and the possibilities for the children's blood type.
Okay, or out of paper.
No, I got you. We can use the paper from last time. I have paper from last time.
You should be adding to your paper that you've already used, and while you're at it, working on it, why don't you tell your bio buddy how many of the practice problems have you done so far?
That's how many have you done?
Me? That's a solid English and Black History packet. How far are you in?
Miss Lynn, can you move a little?
Oh yeah, I'm sorry.
Thank you.
Wait, which A?
Heterozygote A means that I have this A.
Okay, did you set it up something like this? Now, write out your genotype ratios and your phenotype ratios. Genotype ratios and your phenotype ratios.
Genotype and phenotype. Shift right now. You write these as a machine. So 1 to 1 to 1, so there's only one each. And then see how to DNA. So you have a BA blood type, you have an A blood type, and a B blood type as well.
All right, that's all I can stand. Did you get this?
Yeah.
So how many different genotypes did we get? 4. How many different genotypes we got? 4. How many different phenotypes did we get? 4. Okay. Now, this can be used to play like, who's your daddy? Okay. For instance, let's say Susie had a baby. Okay. And the baby's name was Charlie. Okay? And don't say anything out loud. Let's say Susie's blood type is type A. And let's say Charlie's blood type is type O. Okay? Susie, was making some choices. And she had sex with both Jim and John. Okay? Now, Jim has type B blood. And John has type AB blood. So who is not buying diapers? Make a claim and support with evidence. Put it on your paper. Who is not buying diapers? Make a claim and support it with evidence. I'm not saying who is, I'm just saying who isn't. Don't include Charlie as not buying his own diapers. I'm talking about Jim and John. You need to figure it out. This is assuming Susie's being truthful in who she's sleeping with, okay? This is her choice. She made the choice.
Yeah. No, question is, how many Jim was a no? What happens if Jim was a no?
Jim was an O.
Then, no, if John was a no, John was.
Billy was out of the country, so he was not participating.
That's a good question. Johnny, you are not the father.
All right, so let's talk about it. What is Charlie's genotype?
Small eye, small
Okay, they're all eyes. You gotta be more specific. Little eye, little eye. Okay, that is gonna be the genotype for Charlie because that's the only way you can be type O blood. Knowing that Charlie came out of Susie's uterus, okay, that is for sure her son. What would her blood type have to be? This is her phenotype. What's her genotype? Big I, A, and little I. Because that A will give her the type A blood, and if she's a heterozygote, she could give one of these little eyes, right, belong to her. One of these little eyes Charlie got from his mother. Okay, now let's talk about Jim. If we didn't know Jim was having sex with Susie, what are all the possible genotypes for Jim?
Big eye.
Little I, and then
Little I. What's another
big
possible?
I, B, and then IB.
Big eye.
Do you agree with that? Okay. What are all the possible genotypes for John?
Big
Just
eye,
I,
and
just
big
big
eye.
I, A, and big I.
These are his only options, right? Okay. Now, Susie had the child Charlie. We know one of those little eyes came from Susie, so the other little eye had to come from the father. Is John able to provide that little eye?
No.
No. So we know that he is definitely not the father. Okay? Do we know that Jim is the father? No. We would have to do some sort of test, okay? But assuming she only slept with these two gentlemen, we would know for sure Jim's genotype couldn't be what? The homozygous B blood, right? It couldn't be this because we're assuming this little eye is this little eye. Okay? Check with BioBuddy, make sure BioBuddy understands. You would need to do a paternity test to verify that. Okay? Paternity test to verify that.
Yeah, no.
Okay. So we saw, did we still follow the law of dominance? Okay, so it still was big I was dominant over little I, but we had a new thing we rolled in here, and that was what? Codominance, because we had two dominant alleles, and we call those multiple alleles. Okay, we had options on the I. Wasn't just big I and little I. We had big I A and we had big I B. You could have capital T1, capital T2, capital T3, and little t. Right? You could still have dominance, big T, little t, but you could have codominance if you've got two dominant traits that are presenting together. Okay? Now, let's look at another example. Actually, let's go to our notes and make sure they're in our notes. Go to Beyond Mendelian Inheritance. Multiple alleles defined when a gene has many allelic forms or alternative expressions. Many allelic forms or alternative expressions. And I gave you an example there. And then for codominance, when both dominant allele possibilities are expressed. When both dominant allele possibilities are expressed. Youngest BioBuddy, could you review that one time with the oldest BioBuddy? Explaining the difference between codominance, multiple alleles, and how it plays out in inheritance. Go ahead.
So if you have, let's just say hypothetically, the tall one is the dominant, the short one is dominant, it's both expressed and it'd be medium, right? Right? And then multiple allele is when you'd have a lot of choices. because they mask each other. They don't want to be like a straight line. Wait, so what's the other thing did you say?
Next.
All right, moving on. So our next exception to the rule is incomplete dominance. Okay, so if I'm just thinking that through, I want to logically make it make sense to me. So this would be where it looks like you have some sort of intermediate. It looks like blending to the eye. So in this case, you could have a red flower cross with a white flower and get a what? Pink flower. Okay. Now let me differentiate between codominance and incomplete dominance, though they may be one and the same. It's just a little bit of a differentiation. If I have a black bunny crossed with a white bunny, and we have some black hairs and some white hairs, I would call that codominance. Okay? But if I have a black bunny crossed with a white bunny and all the hairs are gray, that would be what? Incomplete dominance. You see the difference there? Okay? If I have some absolute black hairs and some absolute white hairs, okay, that would be codominance. If it looks like blending to me, it would be incomplete dominance. Now from a distance, they probably both look what? Gray. Yeah. Okay? So now you see this in people, okay? Straight hair people are H1, H1, okay? And curly haired people are H2, H2. A wavy haired person is H1, H2. Now, on the way I can remember these is really easy, H for hair, and 1 is a very straight number, and curly is 2 is a curly number. You with me on that? And then wavy is H1, H2. So what I'd like you to do is cross 2 wavy-haired individuals, please, and let's look at their potential offspring. Please give me geno and phenotype ratios, one on top of the other. Genotype and phenotype ratios. Only look up here if you need help. I'm thinking you don't. Geno and phenotype ratios. Okay, we ready? That's all the time I can spare. Okay, what's my genotype ratio? 1, H1, H1. Don't give me any naked ratios. Then what? And? Yeah, if you were doing this in an essay, never give me naked numbers. You need to tell me so I can tell that you know the difference between a geno and a phenotype. Okay, what would be the phenotype? 1 straight hair, 2 wavy, 1 curly. Okay, little tip. When you get a cross, and from that cross you get 3 different phenotypes, it's probably an example of incomplete dominance. And the genotype ratio will match your phenotype ratio, because each genotype expresses its own unique phenotype. Do you see that? Each genotype has its own unique phenotype. Whereas before, if you had straight up dominance, you would expect, if straight hair was dominant over curly hair, you would expect all 3 of these to be what? Straight. And these to be what? curly. But the heterozygote has its own phenotype, and that is wavy. So that's why you get a 1 to 2 to 1 ratio. Oldest bio, is it your turn?
Yep.
Yeah, talk about incomplete dominance. Go ahead.
Okay, so incomplete dominance is rather than new features. Co-dominance, when there's a two of each and it's no longer and then they created a new one when they crossed.
Is this like an explanation of
Okay, now, if you go down to incomplete dominance, it's when a heterozygote has an intermediate phenotype between that of either homozygote. And I gave you an example there, red flower, white flower makes pink flowers. And some examples of diseases in humans of incomplete dominance is hypercholesterolemia and cystic fibrosis. All right, now, penetrance and expressivity. I've talked to you a little bit about that before. Penetrance and expressivity. What do you hear in expressivity? So I'm expressing it in some way. Expressivity must mean I have all, I can express it in a different way. Penetrance, you want to think of this kind of like, how does the genotype penetrate the phenotype? What are the odds, right? And so when you look at this chart, okay, whatever disease you're looking at, this gives you an example. The genotype and the phenotype are both present. So I see the genotype, and what's in the genotype is getting expressed in that phenotype. And they have the disease, okay? In the yellow one, the genotype and the phenotype are absent, meaning they don't have the disease. But in the green, the genotype is present, okay, for the disease, but the phenotype is not there. Okay, so in this case, the dad up here at the top, he has the disease and the mom does not have the disease. And their genotypes and phenotypes show that. Okay? But they had children. How many children? 1, 2, 3, 4, 6 children, yes? Okay, now, when this woman married this man right here, okay, when this woman married this man, she assumed she was marrying somebody who didn't have that disease. Okay, that's the green part. He had the DNA for the disease, but he wasn't expressing it. Okay, so that's talking about penetrance. It didn't play itself out in his phenotype, even though he had the genes for it. So she would have assumed, ooh, I know your dad has that disease and it's genetic. Thank God you don't have it. But then it shows up in their children. Okay, and that has to do with penetrance. Now, expressivity is different. Expressivity is saying, how is it expressed? For instance, if you have syndactyly or polydactyly and you have extra digits, okay, do you have just one extra digit or multiple digits? How is it expressing itself? Okay, and so just to kind of differentiate between that, ping pong back and forth, top to bottom. Youngest, you go first at the first term and then alternate down through those. Go ahead.
Alright, so in complete penetrance, everyone has a single part of that disease has alleles with
Incomplete. Not 100%. Some individuals, they don't have the phenotype. They have the alleles. What happens is they don't express it, it doesn't show up. They're lucky. They think they don't have to, they do in the genes. Right? Polydactyly. You can see it by polydactyly, right? Easily.
And then there's a variable expression. So some people can have this.
All right, so on your notes, incomplete penetrance, dominant trait not always fully expressed. Dominant trait not always fully expressed. An example is polydactyly. You might want to add on, you have little letter I, you might want to be a I, and put expressivity down, so you have that as a reference. So you see the difference between penetrance and expressivity. Add that into your notes. You should be feeling smart, are you? Okay, Miss Lynn. All right, now let's look at another exception to our rule. Okay, so well, what exceptions have we had so far? Multiple alleles, which can lend itself to co-dominance. And we had incomplete dominance. Okay, do you have to have, in order to have co-dominance is even an option, you usually have multiple alleles, right? But for incomplete dominance, do you have to have multiple alleles? No. Okay, so incomplete dominance looks like blending. Okay, then our next one on here is we were talking about penetrance and expressivity. It's not cut and dry whether or not you have that particular phenotype or how that phenotype is expressed. Okay, now our next exception to the rule is pleiotropy. So this is when you have a single gene, but you have multiple traits as a result of that gene. For instance, you could have Down syndrome. Syndrome means it's a whole collection of things that are characteristics of somebody who has Down syndrome. They could have issues with their processing skills. Their tongues are of different, they're a little bit thicker. Their facial features are different. Their fingertips are thinner at the end. Their lifespan is usually shorter. So those are all things associated with Down syndrome. So pleiotrophic effects, a single mutant gene affects two or more distinct and seemingly unrelated traits. Two or more distinct or seemingly unrelated traits. Another place where you see that is sickle cell disease. If you have sickle cell disease, your red blood cells are sickled, so you don't have that biconcave disc all the time, but you have other factors as well. And then one that we'll talk about a little bit is you have, if you are a heterozygote for sickle cell disease, remember you're more resistant to what? Malaria. More resistant to malaria, and that's why that frequency of that allele is so high. And we'll visit that later when we get into our evolution and population genetics. We'll revisit that. Okay, so what do I need you to do there? I think we have everything for that one. Okay, now we're going to look at another exception to the rule. And I want, again, you to think logically so you can distinguish between these. Polygenic inheritance. Poly means what? Yeah, multiple or many. So you look at that part and think multiple or many. And this means genes. Okay? So kind of look at all your options because you kind of have to keep them straight, right? Multiple alleles means I could have big I A, big I B, or what? Little I. Okay? And when we looked at pleiotropy, okay, we said we have one gene, so it could be a gene that's maybe this. But that one gene has several different effects, several different traits that we see. On this one, okay, we have both A's and B's contributing to that one trait. For instance, for intelligence, it's not big S, super smart, and like incomplete dominance. Big S, semi-smart, little s, super stupid. Okay? It's not like that. Your intelligence has S's and R's and Q's and P's and L's and whatever that are contributing to your overall intelligence. Some people are super freaks. They're just like good at everything. They can play 10 instruments, right? They're good at math. They're good at English, write plays, whatever. Okay? They can do everything. Some of you are good, let's say, math and science. Some of you are English and art. Okay? So you can have different strengths, different things that you are good at. That would be an example of a polygenic trait. So I want you to look. What must you have in your polygenic trait in order to be medium brown? What is the qualifier for being medium brown? Look at the three options, and what do they all have in common? Two dominant alleles. Do you see that? To be, or two recessive. To be dark, what do you have to have? Any combination but three dominant alleles, right? To be light, any combination, but you have to have what? To be light, you have to have only one dominant. Do you see that? Okay, so of all your options, there's only one way to be very light, right? There's one way to be very dark. There's two ways that you can be either dark or light, but there are more ways to come up with being medium. Do you agree with that? In dice, this would be like rolling a 7. That is the most frequently rolled number statistically, because that's the one you can get the most different combinations for getting a 7. Does that make sense? Go to Vegas and you'll watch it play out. Okay? That's why you tend to see a bell-shaped curve with polygenic traits. So things like color, because there are multiple ways to be the one in the middle. Okay?
Oh yeah. So are there, for skin color, are there 2 genes and then 2 alleles of each?
Yeah, so you remember you have a homologous pair, right? So you have a homologous pair here and a homologous pair here. So in order to be medium, 2 of these 4 have to be a dominant.
Okay.
And 2 would have to be a recessive. Okay. All right, so you tend to see this bell-shaped curve. Here is in wheat varieties in the coloration, you have all these different varieties. Okay, and then here is height. Okay, we don't just have tall people and short people. We have all different variations in height, and you can see that here they lined up. The women are in white, the men are in blue, and they lined up behind their height. Okay, and so again, you tend to see that bell-shaped curve because it's not a simple, like monohybrid cross that determines your height. So on your notes, you have polygenic inheritance when one trait is governed by 2 or more sets of alleles. And dominant alleles have a quantitative effect on the phenotype. It adds to it. Dominant alleles have a quantitative effect on the phenotype. And 3, the more genes involved, the more bell-shaped the curve. The more genes involved, the more bell-shaped the curve. And your example there is skin color. Now, after skin color, hit a return, and environmental influences will change to D, right? And I want to add in a new C for you, okay? And the new C I want to add in for you is epistasis, okay? Because some reason I took that off your notes. Epistasis. And epistasis means standing upon. It's like one gene rules and impacts the way the other genes are expressed. When one gene interferes with the expression of the others. For instance, when you are an albino. Now I want you to look at the combinations that you see here and this picture and decide which is the epistatic gene, which is the standing upon gene, which gene determines ultimately the color. Okay, discuss with BioBuddy. Look at your different scenarios here. Which is the epistatic gene?
It's B. Wait, whoa. It's actually C. Actually C.
Yeah, it's all small C's.
Oh, and I turned on, we have a new list, Birds of Oak Park. Anybody know who might have come up with this?
Cameron, yes.
Yes. He literally came up with these off the top of his head. He did not consult anything. And so it was quite amazing. He had options. He could have given me more than 40 birds. When you come to me with a list that you want to start, he had more. We had discussions. So then we were deciding what number should we use to log into the list. So we were thinking, I was saying, how about classification? And it turns out, what's the orders of classification? What does it start with? Domain, then what? Kingdom, phylum, class, order, family, genus, species. There are 142 different families of birds, aves. So that's the number, 142 number. 142 number. Oh, I did not know there was a question here. I guess I did put a question in here. 142 number. So tell me which gene, which allele is epistatic. I only need a 1-letter answer.
We'll
What's
see.
wrong?
What?
What's wrong? Oh, I thought you were going, I don't know. Oh, did you disagree?
No,
No, that's
I
it.
don't
Oh,
know.
good.
We'll see.
I don't know, man.
Yeah. Well, I hope.
So, when you look, and I ask you which gene is the epistatic gene, this is what I want you to think about. Because it's obviously about color, right? Because I said an albino. So what do all the colored ones have in common? What do the ones that are albinos have in common? Oh, okay, he's going to go ahead and answer it for us. Okay, so, that's okay. He's like, I will answer that for you. What do all the colored ones have in common? They have at least what? One big C. Okay? Now, as far as what color you're going to be, whether you're going to be black or whether you're going to be brown, that depends on the B part of it, right? But if you have at least one big C, okay, with at least one big B, then you will be the black color. To be brown, you still have to have at least one big C, but you have to be homozygous, little b, little b. You would also know that because, well, black and brown both start with B, so, but little b, and they've lined up with the brown one. So that's homozygous recessive. To be an albino, it doesn't matter about your B-ness. We don't care what B you have. Okay? The whole issue is you are homozygous recessive for the C. So that is the epistatic gene. It's the gene that's in control. Oh, we had three of you passed. Pick the letter C. There are only 3 gongs out there. The rest of you, okay, bells of shame. Bells. Okay, but lowercase c is not epistatic, right?
It's whether or not you have a
Yeah, yeah, whether or not you have color. But if you got the C, you got it. Okay, so did you add in your epistasis? Okay, and what letter is epistasis? C. Okay, I'm just saying. All right, the other thing I want you to know now as well is environmental influences, that the environment can have an impact, multifactorial, on your polygenic traits, And here are some examples of those. So traits, I have trains. Do you have trains?
Yeah.
Okay, watch out for those trains. Traits controlled by polygenes are subject to environmental influences. And then I gave you examples: cleft lip, hypertension, diabetes, schizophrenia, allergies, and cancers. So influenced by your environment. All right? Now, what's our big topic that what is this section titled that we're discussing? What section is this? Look on your notes. What section number are we on? Look at your notes. 11.4. What's the title of 11.4? Beyond Mendelian inheritance. So this is beyond those 3 laws. One item, and the last item we're going to talk about here that's beyond those 3 laws are sex-linked inheritance. And when you look at sex-linked inheritance, who determines the sex of the offspring? The male determines the sex of the offspring. Because a female, every single time, what's the only thing she has to offer? X. She only has the X to offer. But the male could either offer the X or the Y. Okay? And the thing, what it boils down to, is the gene that's on the Y, the Siri gene. And it's the sex-determining region on the Y that is actually what determines whether you will be male or female. So in this first case over here, we have 2 X's. So you would think female, but maybe due to translocation or just any kind of crossing over, you picked up the Siri gene. If you pick up the Siri gene, even though you are XX, and would you have a bar body? If you're XX, yes, you would have a bar body, right? You would still express yourself as male due to the Siri gene. This would be a normal male. You have the X, and you have your Siri gene that would normally be found on your Y chromosome. In this case, you would think you would be male because you have an X and you have a Y chromosome. You have no bar body, right? But you have a deletion that's taken place of your Siri gene, and as a result of not having the Siri gene, in essence, what disease would you have from the diseases that we studied? Not Klinefelter's. This is not Klinefelter's. Turner's. This is Turner syndrome as a result of that. Or
You
the
asked
next
my
question?
question. I was going to
Yeah.
ask about the.
This would present them, this would be phenotypically, this would present it, you would present yourself as somebody who has Turner syndrome. Okay, could I please have the dark-shirted bio buddy explain the Siri gene? Go ahead, dark-shirted bio buddy.
Go ahead. Go ahead.
Oh, okay. So, like usually X and Y to make a guy and XX like a girl.
Siri gene is like what identifies as male.
Okay? So, on your notes, you have Siri gene, sex determining region Y, gene on the Y chromosome that controls development. That controls development. Now, X-linked genes, we're going to be talking about them. X-linked genes that have nothing to do with gender, yet are carried on the X chromosome. Have nothing to do with gender, yet they are carried on the X chromosome. For instance, being colorblind. Now, do you know typically who's more likely to be colorblind, a male or a female? Males. So the reason behind that is this, that there's different types of colorblindness, but your red-green colorblindness is carried on, that's a mutation on the X chromosome. So as a female, how many chances do I have to get a good copy of it?
2.
2. As a male, you've got one X chromosome that you get. If it happens to be the bad one, then you will be colorblind. Okay, and on that note, okay, I need a couple volunteers. There's one, come on up. I need a girl. Okay, get on up here. She's not Jewish, but you're going to marry her today anyway. Okay, Michael. Yeah, is a converter right now. Michael is a boy, okay? So his 23rd pair is going to be what? XY. So there you go, XY. Okay? He is marrying a girl who happens to be a carrier for color blindness. Is she colorblind? No. Okay. They would like to have little girls. Okay? How will they have little girls, and what are the odds that their little girl will be colorblind? Think in your head first. Think in your head first. Think in your head first. A little girl that is colorblind. What are the odds? Okay, so now let's play it out and see if it matches. So what are you going to need to have a little girl? Okay, so the only thing he would, to have a little girl, he would give the X. Now, we have choices, right? Will this daughter be colorblind?
No.
What are the odds of having a colorblind
0%.
daughter? 0%. At best, the little girl could be what? A carrier. Okay? At best, they would not be colorblind at all. Okay? Now, what happens if mom is colorblind? What could we say about their sons and their daughters? Think in your head first, what you would come up with in your head first. Not out loud. You guys immediately start talking. Think in your head. I want you to come up with it on your own. Okay, now share with Bio Buddy. Sorry, your Bio Buddy got married. Okay. Let's make little girls again. Go ahead, make a little girl. What's going to happen to the little girls? Carriers. Will they have the disease? No, but they will all be carriers. Now let's make little boys. What's going to happen? Colorblind. Who did they inherit the color blindness from? From the mom. Okay, now, let's see, we'll go like this. Now, what could possibly happen? I want Blue to talk about the daughters and Slate to talk about the sons. What could possibly happen? You guys can talk to each other. Wait, show them so they can see it.
Oh, okay.
No, it's possible.
Oh, yeah, this next section.
You're all. Okay, let's have little girls first. So what are our options on our little girls? You would predict that half of the little girls would be what? Colorblind, and half would be what? Carriers. Okay, let's have little boys. What's going to happen? What?
Nothing.
All the boys? Half the boys would be fine, and the other half would be carriers. Can little boys be carriers? They'd be colorblind. Did we already do where she had colorblindness? Yes. Okay. Now, keep that. Blue, talk about daughters. Slate, talk about sons.
Oh, yeah, no, carriers.
No, daughters will be carriers. But daughters are carriers.
Okay, let's have little girls first. Okay, what's this one going to be? Okay, and what's the other option? Okay, carrier both ways? Yes. Okay, what about little boys? Yeah, we don't care. The dad is never going to pass on his sex-linked diseases to his sons. Why is that? He will always give Y to his sons. Now, riddle me this. What could we say if we didn't know, if they were hiding their genotypes? What could you say about the parents? Hide your genotypes. What could you say about these parents if they have a colorblind daughter? What could you say? Discuss with your bio buddy. What could we say the genotypes are if they have a colorblind daughter? What are the possibilities? Okay, here, you make it. If you have a colorblind daughter, what are your possibilities?
They both have to be.
One color, yeah, you have a colorblind daughter.
Yeah, she could. She just can't.
Okay, what do they need to get? Is this right for the dad?
Yes.
If they have a colorblind daughter, he has to give a bad X. You know, if all their daughters are colorblind, he's going to give a bad X every time. Okay? If all their daughters are colorblind, now, or just any daughter is colorblind, she could be this genotype, right? But what else? She would be colorblind as well. Or what else could she be? She could be a carrier, and they could still have a colorblind daughter. So when we do Punnett squares now, okay, when we do Punnett squares now, set yourselves up like a Punnett square. Okay, is that how we would do it? There we go. Okay, when you do Punnett squares now, you will need to put your letters on your Punnett squares, and then as a superscript, you're going to put on there if it's what kind of trait it is, and you have to add it to the X's because you need to see it in the offspring. Okay, so thank you, good job. You're divorced now. I'm sorry. It's a great marriage. When you do a Punnett square, you have to write what the trait is on the superscript, and I'll give you an example. We're going to practice that in just 1 second. This work, go to your notes on this one. Okay, Thomas Hunt Morgan, if you look at that right there, and fruit flies. And the reason he discovered this with fruit flies, they're really simple. There are only 4 homologous pairs for fruit flies, so they were easy to study. And he looked at traits that are dominant and recessive and sex-linked and mutant traits, normal wings versus vestigial wings, and when he did crosses in order to study them. And if you look at the males and females on fruit flies, they actually physically look different. Not that you've ever studied a fruit fly up close, but you could. Okay. And Blue tells Slate, what are some differences between male and female fruit flies when you look at them structurally? Who has more stripes? Who has a pointy abdomen? The females. Do you see how it comes to a point right here and we're rounded here? Okay, and they have sex chromes on the males, and you can see differences. So you could do these crosses, and he did hundreds of crosses with different traits in order to figure this out. Okay, reminder again, you already know this. So let's set up one of those crosses. Okay, so we have red eyes. Eye color is carried on the X chromosome. Red eyes are the wild type, or normal, wild or normal, and white eyes are mutant. All of the sex-linked traits that I'm going to give you, the mutations, are always going to be recessive traits. They aren't in real life. There are dominant sex-linked traits. I won't give you any. And for starters, we want to cross out a female who has red eyes. And she's going to be homozygous red. So she's X big R, X big R. Now, he has white eyes. So what do you think his genotype is going to be if he has white eyes? Okay, not eyes. You're not you. We don't have big X's and little X's. X little r y. Okay.
Oh, so are they all?
Okay.
No idea.
Yeah, you're always going to put your letters on top of that X chromosome, and you don't have to worry about the Y. There are traits on the Y chromosome. They're called holandric traits when they're found on the Y chromosome. But remember, I gave you an example like that. That's like hairy pinna, having hair sticking out of your ears. That's something that's found just on the Y chromosome. Okay, so if this female right here is crossed with this male, okay, so if we put the female here and we crossed it with this male, what would this be? It would be a what? Yeah, it would be a red female. Okay, now do all of your crosses.
FF.
Oh my god, I get it. Dude, because the okay, I'm stupid. I'm so stupid.
Red eyes is dominant over white.
As men as an expressive. My thought just got oh my god.
It would be. There's nothing on the Y. You don't have to worry about the Y. Nothing that there's nothing that we're concerned about right now. There are traits on there, but nothing we're concerned about. The eye color is
Because
on
of
the
little
X.
r. Because I mean.
Okay, the same.
Big
the same
T,
trait.
small
The
t,
same
yeah.
trait. Okay. Got it.
Okay, so I'm going to switch because this is a little bit better to see. What would this one be? X what? Big R, then what?
X little.
X little r. So what would this express itself as?
Red-eyed female.
A red-eyed female. Good. This one would be X big R and Y. This would be a what? A red-eyed male. And this would be what?
Same thing.
X big R, Y, and this would be a red male. Okay, how many different types of females are there? How many different types of females did we see in this Punnett square? One. Okay, so this is the parent generation right here. This is the parent generation. These right here would be referred to as the what? F1s. Now, there's only one type of female here, right? And only one type of male here. But everybody has red eyes, yes? Okay, so here, when you got your F1s, which is what we have in here, this red eye right here, the females would be X big R, X little r, and the males would all be X big R, Y. Do you agree with that? Now let's cross the F1s and make some F2s. So cross these F1s in red that I wrote up there, and now do your Punnett square and make some F2s. Only look up here if you need a little help. Okay, so now we're doing an F2 cross. Crossing the F1s to get the F2s. Okay, so when I do this, this would be in black now I'm writing for my F2s. What would this be? A red female. This would be a what? Red female. And this would be a red male. And this would be a what? Okay, do you see how we do that? Did we put anything on the Y? No, we didn't put anything on the Y. Are there traits on the Y chromosome? Yes, but we're following a trait that's on the X. That's why we're calling it an X-linked trait. So we're only putting letters on the X because it is an X-linked trait. Okay? So when you do, when you look at a pedigree, okay, because all of the X-linked traits that I'm going to give you are recessive, when you look at a pedigree and you're trying to identify, is this a dominant trait? Is this a recessive, autosomal dominant, autosomal recessive, or is it an X-linked trait? When you analyze a pedigree, since all the sex-linked ones I'm going to give you are recessive, you're going to know that in your head, right? Okay, so you would expect for an X-linked trait that you could skip generations just like you could in an autosomal recessive, right? On dominance, we don't skip traits, right? Because you can't mask it. Unless you have an issue with what? Learned about it today. Penetrance, right? Penetrance, okay? You're not going to skip generations. So here, we're skipping generations. So I know this is either an autosomal recessive trait or it's going to be an X-linked trait because it's skipping generations. Now, why would I guess that it is an X-linked trait? Because it's presenting itself more in what? Males than females. Who's the only one who has the trait on here? Only the males. And you could predict their genotypes, right? What would be the genotype of this individual right here? If we were sticking with the letter R's, what would be the genotype here? X little RY. And this one would be what? At least one big R. And then do we know what the second one is? No, we're not 100% sure. Okay? But you could work your way down and you could go, okay, well, he had this daughter. Do we know her genotype? What's her genotype? You know she has a little r because of who? The dad. But you also know she has a big R because she doesn't have the trait. Who did she get that from? Her mom. She got the big R from the mom. She got the little r. That's all he has to give if he's having a daughter, right? Okay. And then she married a man. Do we know his genotype? No, we don't? He has to be big R because he doesn't have the disease, right? See how we're working it?
Yeah.
Okay. Do we know about their children? What's this little boy? Right? Because he has the disease, and he's a boy, so we had to get the Y chromosome from dad, right? And he, just the luck of the draw, 50-50 shot that he would get the bad X. Okay, do we know what this little girl is? We know she has at least one big R, A, because she doesn't have the disease, and B, because that's all dad has to give. Do we know what her second letter is? It could be big R or it could be little r. We don't know. Okay? And what I want you to be able to do, okay, is I want you to be able to look and capture this and put it in your notes. I want you to be able to look, I'm asking you as a question, would this show up as a dominant trait? Which of these pedigrees would work? This could be a map. But I also want to know, could it show up as a recessive trait? Could it show up as an X-linked trait? So for each one of these sample pedigrees that I gave you, decide, You know, would it work for dominant? Would it work for a recessive trait? Each one. Would it work for an X-linked trait? Okay, and I'm not going to take the time to work those out because I want you to think about it. Okay? I want you to try to work through it. If somebody puts it in your notes, put it in the bottom, like check answer, put it, you know, at the very bottom so it's not obvious, so everybody can look and figure it out for themselves. And then you can have a little discussion about whether you think it's right or wrong in your group share notes, and I will go over it with you next class as well. Okay, or at the review, more than likely. I'll do it at the review. Okay, so things like when you look at, everybody have it? Okay, don't work on it right now. I want your attention up here. Okay, when you look at a pedigree, usually what you see is that you have more males than females that are affected. A son can have parents who have the normal phenotype, and we saw that in our little marriage we had going up here, right? So the son could have it even though neither parent presented as it. But when a son gets it, what do you know about the parents? Okay, dads can't be carriers, so be more specific. Mom had to be, right? Okay. And then for a female to have the characteristic, if a female has it, her father has to have it. Because she has to get 2 bad copies. One of those bad copies is definitely coming from dad. Okay, so he definitely has the disease. It can skip a generation because we're going to be only looking at recessive sex-linked diseases. And if a woman has the characteristic, all of her sons will have it. If a woman has an X-linked disease, for sure all of her sons would have it. That would be why you might go and see a geneticist. You might choose to adopt, or you might choose to look at doing sperm, or not sperm selection, looking at the egg afterwards, you know, when it's to see whether or not it might have the copy of the good gene or a copy of the bad gene if you're a carrier for it. These are all things people consider depending on what kind of disease they have. Color blindness is often talked about with sex-linked. They use the letter B, which is kind of annoying because that would imply that being blind is a dominant trait, colorblind, but it's not. It's a recessive trait. So when you see that, if you look here, X big B, X big B, X big B, okay. That would be a normal female, where this one, don't even look over there. Just look here. That would be a what? Carrier, right? This would be somebody who has colorblind, normal male, and colorblind. Males can't be carriers because they only have one. They can't hide any of those traits. Okay? And do I have anybody that's colorblind in this room? Okay, let's see how he does. Okay? Log in your answers. That's awesome. Don't say anything out loud.
We just did this. He showed me a picture online. You couldn't do
Oh,
it.
yeah?
Come on, just focus. You could do it. I believe in you.
What's the answer?
I can't give you the answer. I'm sorry, man. I need you to think though.
60.
Yeah, you write it up. You have to have 40 options.
Try.
And then when I'm not on my
Show me the dark
computer, you can
spots.
type it in. Like, you know, lunch
The dark
period when
spots.
I'm not on my
In
computer.
the middle. Just look at the middle. It's central.
Let's go, Thrasher.
Here's what I say.
What's the answer?
Put this down. Yeah, let's do it.
If you don't see anything, you can
Oh yeah, let's do it.
put in what you think you see.
It looks like a 2 on it. It looks like an S, backwards S. But I'll put 8. If you say it's 8, I'll put it.
Uh-oh, somebody got it wrong.
Or was it Max?
All right.
Delete it.
So,
Wow. You
red, green, colorblind, see the number what? 3. What did he get? 3. Okay? Wow, I knew that ahead of time, okay? So that is what he saw, okay? Which
I
is
see
pretty
3, you
awesome
said
saw.
8.
Want to do another one?
Yeah. Oh, rip.
This one, I don't think I have the answer for this one.
Max, I want you to focus.
The last one was way easier, bro.
You can do it.
Max, think. Max, just focus. Just focus. Try.
How much? 10.
Try.
Think.
9, 8.
Is a
Before you put it in
Jedi
the
warrior,
just
it's
try.
true or not true.
No. No way.
You don't see it? Okay, yeah. What number is it?
Not seeing 5.
5.
bro.
Let's do another one.
Easier, easier.
Yeah, easier.
Don't say it. Don't say it out loud. Let him struggle. The struggle is real. Oh, it will.
Be okay.
How do you guys like see these?
Do you want to get a 50% on the quiz?
Oh, I'm gonna look at Yeah.
Did you put a number in? Go for it. What do you see?
A number.
13. Is it 13?
It's not 13.
Put in whatever you don't tell
He
him.
looked at my clicker. I'm hiding it now.
Oh,
Yeah.
he looked at your clicker.
Yeah.
Okay. Yeah. Yeah, red greens usually see 17, or total colorblinds don't see any number or a different number. Yeah. Okay.
See any numbers?
Now, the first one was best. Okay. So, there are, catch this. I don't want to take the time to do it right now because I want to introduce the next chapter for you, but this is one that you could do. There are practice problems in your book. Capture it. You captured it? You can get it out of the PDF too.
Colorblind?
Yeah.
Excellent.
Can you do it in your head? A normal male with a female who is a carrier. Let's do it together real quick. Make sure you're okay with it. How would I set this one up? A normal male. So what would a normal male be? X, big B, Y. Okay, and female who's a carrier. How do I set that up? All right. So this would be what? X big B, X big B. What would that be? Yeah. Female, and she's what? Normal.
Oh, she's a carrier.
And, okay,
I
guys.
didn't see carrier.
Ori, we're right here. X big B, X little b, what would that be? Female carrier. So, any daughters with a chance of being colorblind? No. No daughters with a chance of being colorblind. Now, any daughters with a chance of being a carrier? Yeah, okay. Then let's do our boys. X big B, Y. What would that be? Normal male. And X little b, Y, what would that be? Colorblind. So any chance of sons being colorblind? Colorblind? Yes. What are the chances of a son being colorblind? 50%. If I said, what are the chances of any child, you would tell me 25. But if I ask you any chance of males, then you're going to tell me 50%. Because you would only look at the male offspring. Do you see that? Okay. All right. And then things like Duchenne's muscular dystrophy is a sex-linked disease. And there are other options as well. I didn't put this in your notes, but you can have fragile X, and literally, I don't know if you can see it. See how part of the X is coming off right down there? And it is the most common form of mental retardation. Children are hyperactive or autistic, delayed speech. They can have facial features, other issues that go along with that, but that is on the X chromosome. Okay, and then there are lots of practice problems that you could do. And then the last part, I don't know why I don't have this on here, where I must have cut it off, but it's got to be in here somewhere. Environment and phenotype. On this, you have a Himalayan rabbit, and what they did is they shaved the back of the Himalayan rabbit and put an ice pack on his back, and then when the hair grew back in, what color did it grow back in as? Black. So here is where and remember when I talked to you about how the environment could affect those traits, your multifactorial, your polygenic traits? Okay, so this is an example where the environment could influence it. And in this case, the hair is growing back black in that area. Can you think of why this would be a good adaptation? Yes.
Capture the sun's energy.
Yeah. So what are the coldest part probably on the bunny? First of all, why would the bunny even want to be white? To blend in with the snow, right? So what are the coldest parts that he would be exposed to would be the snow on the ground, right? And then his thin little ears poking up, right? So if those parts of the bunny were black, they would absorb more wavelengths of light. Does that make sense? So that'd be why it's adaptive. Maybe that's why it grows back black there. When you look at reptiles and whether they're males or females, it has to do with the temperature of the nest. Temperature of the nest. Now, somehow the females know, and they may want to produce more males or females depending on how deep they dig their nest. Will vary the temperature of the nest and the offspring they will generate. And then this brings into case nature versus nurture. How much of our behavior is a result of our nature, what's in our DNA, and how much of it has to do our behavior with our nurturing. And we'll talk about that when we do the behavior chapter. Okay, and then I cannot say enough. Do your practice problems. Okay, they are embedded throughout the chapter. They are at the end of the chapter. You can go online and do even more practice problems as well. Okay, and if you're watching this late at night, have a piece of, see, you all watch late at night, and make good choices. Don't be bad choice Barbie. We really love
Oh my God,
Matt,
it's so good.
even though he's colorblind.